everybody. So this is a side project of our early career award, and shown here are just the authors' pictures. How do I advance? Oh, there we go. How do I advance? There. Oh, oops, that's weird. Okay, so sorry about that. So biofilms are communities of organisms that are held together by a self-produced extracellular matrix, and they're all around us, and for good and bad, they're part of our everyday lives. So on the left are some beneficial biofilms, including in the treatment of wastewater and our gut microbiome, and these are very important for protecting us. However, on the right are some detrimental biofilms. So on, you can see that their biofilm is formed on a pipe, and this leads to about $2 trillion a year of damage due to the corrosion from that biofilm. And biofilms can also cause disease, and about 80% of infections are thought to be caused by biofilms. So the problem with detrimental biofilms is that the cells in that biofilm are much more resilient than their free-swimming counterparts. Part of that resilience is due to this matrix material that holds those cells together. So this includes exopolysaccharides, extracellular DNA, lipid vesicles, and membrane and proteins. My lab previously, using the model biofilm organism, Pseudomonas Aeruginosa, identified proteins in this biofilm matrix, and one of those proteins is called Oprf. It is an OmpA-family member and an outer membrane porin that's conserved in about 5,000 gram-negative species. And we're interested in this protein because it's been identified in the matrix of many different organisms, biofilm matrix of multiple organisms, and particularly found to be abundant in membrane vesicles. So we made a mutant of removing the gene encoding this protein, and using a standard static biofilm assay, we found that it actually produces less biofilm than wild type. So you can see that in this assay we've normalized to wild type, and our Oprf mutant makes more biofilm than our negative control, which lacks the ability to make EPS. However, it's about 40 to 60% less than wild type. And so this is at the 24-hour stage of development, so we then wondered, well, when does this actually occur? And so down here on the y-axis is relative biofilm biomass just for the Oprf mutant, and it's normalized to wild type within the same time point. And so you can see for the first 16 hours there's no effect, and it's only really in late biofilm that we see this defect. And so this strongly suggests that Oprf is involved in the maintenance of the biofilm. And this is interesting because now Oprf, you can think about it as a target for both promoting biofilms to stay where we want them to stay and maybe getting rid of them where we want them to leave. And so if you're interested in the mechanisms behind this, I am poster 9 on Wednesday, and so we can talk more about it then.